Hello, everyone. In this session, we're going to dive into the roots of modern biology by exploring Mendelian genetics. We'll look at how a monk in a garden uncovered the basic rules of how life passes traits from one generation to the next. It all started with Gregor Mendel. He's often called the father of genetics because he was the first to rigorously study inheritance, or heredity, which is just the passing of traits to the next generation.
Mendel worked with garden peas. Now, peas usually self-fertilized, but Mendel wanted control. He meticulously cross-pollinated them by hand to see exactly what would happen when he mixed specific traits.
Let's look at one of his famous experiments. He took plants that always produced yellow seeds and crossed them with plants that always produced green seeds. He called this the pea or parent generation.
Now, you might expect a blend, right? Maybe yellowish-green? But no. The offspring, which we call the F1 generation, were all yellow. The green traits seemed to have vanished completely.
But Mendel didn't stop there. He let those F1 plants self-fertilize to create the F2 generation. And look what happened. The green trait came back. It appeared in a specific ratio, 3 yellow plants for every 1 green plant. This 3-to-1 ratio was the key. It told Mendel that the green trait hadn't been destroyed. It was just hidden. Mendel realized traits are controlled by factors, which we now call genes, but genes come in different versions. We call these alternative forms alleles. For example, the gene for seed color has an allele for yellow and an allele for green. They are different versions of the same instruction set. This brings us to dominance. Mendel saw that some alleles can mask others. The dominant allele, like yellow seed color, shows up even if only one is present. The recessive allele, like green, only shows up if the dominant one is missing. We use capital letters for dominant alleles and lowercase for recessive. So a plant with big Y, little y will look yellow because the yellow is dominant. We have special terms for these combinations. If an organism has 2 of the same alleles, like 2 big Ys or 2 little y's, it is homozygous. If it has 2 different alleles, it is heterozygous. You'll often hear heterozygous organisms referred to as hybrids.
It is crucial to distinguish between what genes you have and what you actually look like. Genotype is the organism's allele pairs, the code inside. Phenotype is the outward appearance.
Notice this. 2 different genotypes, big Y, and big Y, little y, can result in the exact same phenotype. You can't always tell a plant's genetics just by looking at it. Mendel summarized his findings in laws. The law of segregation states that the 2 alleles for a trait separate during meiosis. They don't stick together. Each parent passes only 1 allele to their offspring. Then there is the law of independent assortment. This says that genes for different traits, like seed color versus seed shape, sort independently. Being yellow doesn't force a seed to be round. It's a random mix. To predict these outcomes, we use a Punnett square. Let's look at a monohybrid cross, a cross involving just 1 trait. If we cross 2 hybrids, big F, little f, we fill the boxes. We get 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. This gives us that famous 1-to-2-to-1 genotypic ratio, but physically, it's a 3-to-1 phenotypic ratio, just like Mendel saw in his garden. Things get more complex with a dihybrid cross, where we track 2 traits at once. Now we have 4 types of alleles from each parent. This huge grid helps us calculate probability. While real life is messy and random like a coin flip, with enough data, the offspring ratios usually match these predictions closely. So from pea plants to probability, Mendel established the framework for how life persists. He showed us that inheritance isn't magic, it's math and biology working in concert.